My name is Cindy Chestek. I am one of Luis's two co-advisors. What you're going to see today is very much a mix of his work with Chandramouli Krishnan. That'll be like the first two papers. The thesis ended up very 50-50, into more brain-machine interfaces work. But I just want to take a minute to introduce Luis. So he came to us with a degree in robotics and computer science. So already like taking a risk on this new robotics discipline. And so I think I want to introduce him first, both sort of on a personal note, and then let me introduce the note, the thesis a bit. So first, on a personal note, Luis has just been an absolute pleasure to have in the lab. I have to say, I'm speaking for maybe his lab mates a bit here, but I think they won the lab mate lottery on having one of the most helpful people in the lab that has had a finger in so many different projects and is just wonderful to work with. Interestingly, he buys birthday balloons for people in the lab on their birthday. The catch is that they are never happy birthday balloons. So they're always like, get well soon, or like, it's a boy, or something like that. So absolute pleasure to have him in the lab. On the science side, so what you're gonna see, so first of all, excellent work in rehab robotics that, is much more, Chandramouli's expertise. Let me say a few words on the last project he's gonna present on his NeurIPS paper. Deep learning is kind of taking over the world, and we're seeing it all over the place, but there's maybe less of it in this robotics building because they're scary black boxes, right? And we're much more comfortable with maybe a more controls, deterministic approach. So what you're going to see in the last AMP presents is what I think is a glimpse of the future in which neural networks are not black boxes anymore, and we're starting to combine them and incorporate them with a more deterministic controls approach. So with that, I will let Luis take it away.
Thank you, Cindy, for that very kind introduction, and thank you all for being here, both in person and a bunch of people from Chile and other parts in Zoom as well. I really thank you for being here. So today I'm going to present my thesis, and the title for that, it's a mouthful, is Understanding Motor Control for Assistive Technologies from Biomechanics to Brain-Machine Interfaces. Okay, there you go. So humans are amazing machines that you can think of as having similar attributes to robots. We have perception to sense the world around us through our eyes, sensors in our joints tell us where our limbs are, receptors in our skin let us sense pain, touch, temperature, and then we can reason about that information through our central nervous system, through our brain, our spinal cord, and then we can act on the world through motor control by moving, for example, our limbs. Now, humans are a lot better than robots are at this motor control part. So if we can understand well how humans do motor control, then first we can develop better machines that potentially work more as humans do, and then also potentially develop better assistive devices for those people in which some part of the system is malfunctioning. And this idea is the one that has sort of driven most of the work that I've done during my PhD. Now, during the PhD, as Cindy was saying, I've had a chance to work on pretty sort of broad topics, and I want to split my presentation today into three. First, I'm going to talk about biomechanics of ankle motor control, and I'm going to be more specific and talk about my work on the reliability of ankles' mechanical impedance parameters. Then I'm going to talk about motor learning for extending motor control. And I'm going to talk about our work on designing a novel web-based platform for designing sequential motor learning experiments. And then I'm going to talk about our work in brain-machine interfaces for restoring motor control, and more specifically on our work on explainable algorithms in brain-machine interfaces. So the first topic for today is the biomechanics of ankle control. And here I'll talk about our work, a paper that we did on the reliability of the ankle impedance parameters. And this was actually published about a year ago in the Journal of Gait and Posture. So one strategy that the brain may use to achieve good motor control is to modulate the mechanical properties of the joints. And we see this on sort of everyday examples. If we're playing baseball, if you do, and when you go to catch a ball, then you make your shoulder, your elbow, your wrist a little bit more stiff so that you can properly catch the ball. This happens also when we're walking. We make our ankle more stiff as we touch on the ground so that we can store elastic energy and then release it as we lift off. And that allows us to walk more efficiently. Now, these sets of mechanical properties are referred to as the joint impedance, which is defined as the resistance to motion when force is applied. And this joint impedance is typically modeled as a second-order system in which we have a stiffness component that resists position changes, a viscosity component that resists velocity changes, and then a mass or inertia component that resists acceleration changes. Now, the ankle is one of the most important joints during walking, and thus it has been widely studied, like the ankle impedance during walking and standing has been widely studied. So to do this, what studies have done is apply sort of a system identification approach in which they perturb the ankle and then they measure the response, and then they can evaluate the mechanical properties of the joint at that point. And this technique of the system identification technique has been used to compute the ankle's impedance parameters during standing and also during walking. So what I'm showing here on the right is a plot that shows in the x-axis the percent of stance phase. The stance phase is the part of the walking where your foot is actually touching the ground. So you can see that as you sort of move along in the stance phase of the gait cycle, you increase your stiffness, and this shows this modulation of impedance, in this particular case, stiffness throughout that stance phase. Now, why do we really care about the ankle impedance and how the brain is modulating it over time? And the first reason for why we study this is because it can be potentially used as a clinical metric for tracking progress on patients. So what I'm showing on the right is a study from a few years ago, and it's showing in green patients that had had a stroke, and then in gray, healthy adults. And you can see that there's a difference in how they modulate the impedance throughout the stance phase. So you can potentially use measuring the impedance of the ankle as a way to determine how close or how far from baseline a certain person is, and then design interventions that actually bring them closer to that baseline. And then the second reason for studying this is because understanding how the ankle impedance goes over time can be a biomimetic target for prosthetic control, in which we can design prosthetics that work more like human ankle, like the human ankle does, and potentially walk more naturally and even more efficiently. However, it's unclear how reliable this method is in sort of this system identification method in measuring the ankle impedance. And if it's not reliable, we may draw the wrong conclusions from it. So what we did in this work, we tested the retest reliability of this method in measuring the ankle impedance during standing and walking. And we also measured the minimal detectable change, which is sort of the measurement error of the system. And I'll get into that later as well. So to do this, what we did is we applied small perturbations to the ankle on participants that came in during standing and walking. And to do that, we use what is called the perturbator robot, which was developed by Professor Rouse here during his PhD. And you can see just part of it on the image on the right. And after applying these perturbations, we measure the effect or the response of the ankle, both in terms of ankle angle and ankle torque. The ankle angle we measure with motion capture, and you can see this reflecting markers on this participant legs. This is me, is that participant. I am that participant. And we use that plus the force platform, the measurements that we get from a force platform, to estimate the ankle torque as well. And then having that ankle torque and ankle angle, we fed a second order model similar to what I showed before, that is composed of stiffness, viscosity, and inertia. So overall, for data collection, we had exactly 18 participants that came into the lab to do 2 sessions, test and retest, each about 3 hours long. So it was a big time commitment. And then we had participants do standing and walking perturbations. Standing perturbations are sort of what you can see on the GIF there. They're very small. And then walking perturbations are very similar, but they are applied at specific points in the gait cycle. So here are the results that we found for stiffness, viscosity, and inertia for the 2 sessions. Now, if this method was perfectly reliable, then we would see these 2 measurements being very highly correlated and also to agree a lot to each other. And the way we measure this is with the ICC, or intra-class correlation coefficient, that measures, again, how correlated these two values are and how much they agree with each other. And we interpreted those values using a previous study, and we find that overall stiffness, viscosity, and inertia had good to excellent reliability. We also measured the MDC, or minimal detectable change, and that's the last column over there. And what that tells us is a little bit about the measurement error of the system. So what we found here, for example, for the stiffness, we see that it's a value of 0.81. Then that means that if you're designing an intervention and you want to evaluate whether that intervention actually changes the stiffness of the ankle, and you see a change after applying that intervention of less than 0.81, then you would not be able to say that that intervention actually changed the ankle stiffness. And so it can be a valuable sort of reference for people designing studies to use this as an outcome. And then we did the same for walking, and we find that the reliability of stiffness and viscosity was excellent, but the reliability for inertia was only fair. And then I'm showing this in a different way for walking. What I'm showing on the x-axis is the value for the test session, and then in the y-axis, the value for the retest session. And I'm showing the stiffness on the left and then viscosity on the right. And I'm also showing two cases, one in which we removed the impedance of the robot, which is shown in blue, and one in which we did not, which is shown in orange. And overall, if the measurement, if this device was sort of perfectly reliable, we would see all these data points lying exactly on that black segmented line that you can see on the back there. And what we see is overall, we're pretty close to that. And they're also very correlated to each other, as you can see by the R values, which represent the Pearson correlation coefficient. So overall, we found high reliability for measuring stiffness and viscosity, both during standing and walking. And this suggests that they can potentially be useful for tracking clinical progress, as well as for designing better control algorithms for prosthetics. And then second, the minimal detectable changes that we report here can potentially guide the interpretation of interventions that actually use these values as the outcome. So now switching gears a little bit, I'm going to talk about motor learning for extending motor control. And more specifically, I'll talk about our work on a novel open source platform for running online sequential motor learning experiments. And this paper was published almost 2 years ago now in the Journal of Computers in Biology and Medicine. So motor learning, for those not familiar with it, is the field that studies how humans improve their motor control. And it is typically categorized into two: motor adaptation and motor skill acquisition. Motor adaptation is thought of as a more shorter term process. So let's say you know how to drive your car, but then you go into someone else's car, and that new car has a little bit different sensitivity on the accelerator and the brake pedal. So you have to adapt your motor control to that new car. And you can drive the new car similar to what you did with your previous car. But then when you go back to your car, you don't lose that, and you can return to baseline and still be able to drive your own car. Motor skill acquisition, on the other hand, is thought of as a longer-term process, and in which you're acquiring a novel motor skill. One novel and very cool motor skill, in my opinion, is that one-handed backhand by Roger that I don't think I can learn to do. So to study how humans learn, we cannot have people do that one-handed backhand because you're going to run into a lot of issues. So motor learning experiments usually simplify the experiment design to sort of test specific hypotheses. And one paradigm that is very common in motor learning are sequential motor learning experiments, in which they have participants do like a sequence as fast as they can, usually, of characters, or as accurately as they can. And then what they do is they measure speed as the outcome. That's sort of what signifies learning. And this is a very common plot. What I'm showing here on the bottom is a very common plot in motor learning studies, in which you see this very fast improvement at the beginning. And what I'm showing in the y-axis here is tapping speed. So they're getting faster over time, and then they sort of converge to final velocity. And sequential motor learning experiments are cool and interesting because they require very little specialized hardware, and also because they're very simple overall. Now, the need for bigger and more representative samples of the population to study motor learning in a sort of broader sense has motivated researchers to conduct web-based or online studies. And this sort of also goes in line with the COVID pandemic we sort of still have, that what made it impossible for participants to come to the lab to do experiments, right? And there are some very cool tools that have been developed for this purpose to allow researchers to conduct these online, to create and then actually sort of distribute these online experiments. These are just some examples: Lab.js, PsychoPy, and then Gorilla. And they're really, really cool. They're very flexible. They allow you to create very interesting experiments. But the problem is the modern learning field is very interdisciplinary. And that means that a lot of the people that want to research these scientific questions don't have the technical skills that are required to actually use these tools, because that flexibility comes with the need for coding most of the time, actually. So we wanted to build a platform that catered to these non-technical researchers. And we created a platform that that didn't require sort of coding skills. So in this work, we built the platform. We validated it by replicating a previous study on microlearning. And then we also showed its ability to sort of explore new scientific questions by studying skill transfer learning between limbs. So we designed the application to cater to two types of users. First, the researcher, and then second, the participant. The researcher can go into the site, which I showed below, can log into their profile. They can create one if they don't have one. Then they can see what studies they have unpublished or published. Published meaning that they have actually started distributing and collecting responses from. And then they can see for all those how many responses they have, and then download the data if they want to do further analysis. Then they can create new studies, and within those studies, they can create new experiments. And we chose a form-like interface for creating experiment. And this doesn't allow the same flexibility that the other tools that I mentioned sort of provide, but it allows non-technical researchers to create experiments very easily and very fast. Then the researcher can go and test that experiment, and this is how the experiments look like in the platform. You can see that sort of sequence of characters, and you have to type them usually as fast as you can. Then when the researcher is ready, like they decided that this experiment is the one that they want to actually start collecting data on, and they can distribute it, they can send out emails or any other sort of form of communication, and then the participant can enter the site, enter the experiment code and group code potentially of the experiment that they want to be a part of, and then they can just start doing the experiment. And after they finish the experiment, the data gets automatically uploaded, and then the researcher can see on their side that there's a new response on your experiment, and then download the data of that participant as well. So to validate that experiments could actually be done, so like this is a valid scientific tool, and we replicated a previous study on microlearning, or how much you can learn with very short periods of practice and rest. And so we designed an experiment that followed the same paradigm as them, but we added a small part which I'll touch on in a little bit. And so we had participants do 2 sets of 36 blocks. In each block, they had to do the sequence first with the left hand, 4, 1, 3, 2, 4, as many times as they could within a 10-second block, and then they had 10 seconds to rest. And then the same for the right hand after the first 36 blocks, in which they had to do the sequence 7, 0, 8, 9, 7 as fast as they could. And this is sort of following the paradigm that that study on microlearning was looking at. We also wanted to test whether we could just share this platform for participants to do it in the comfort of their homes, or if they would sort of see some problems and see some differences in how they do the experiment in the lab versus how they do it at home. So we collected 2 groups of participants. First, 18 supervised participants in which they came to the lab, and we had someone from the group looking over their shoulders, basically making sure that they were following the instructions as they should. And then we also had 25 unsupervised participants in which we just sent out the experiment, and they did it with their laptops and from the comfort of their homes. And I wanted to take a moment to thank, because a lot of people here actually participated in this experiment, so I thank you for being a part of science. So this is how the experiment actually looked like. They had a sort of a progress bar in which they could see what block they were at. They could see what sequence they had to type. what hand they had to use, and then they sort of started typing the sequence. So as they typed the keys on their keyboard, the corresponding keys lighted up. In this case, we're showing sort of the same color for every type, every time they type the same color, because we were not interested in seeing sort of how they react to errors, but you can change that if you wanted to in our platform as well. So what I'm showing here is some results comparing the supervised and unsupervised groups for the left-hand blocks and then for the right-hand blocks on each side. And you can see that overall the two groups are very similar, and in fact we found no differences between the two groups. They had similar progress, they achieved sort of similar tapping speeds, and they started similarly as well. Something interesting that we found, and I'm going to get to more into this later, but they started doing the blocks of the left hand slower than they did on the right hand, as you can see sort of from the beginning of the two plots. And I'll get into that later. So the study that we were trying to replicate was looking at what happens during this sort of micro scale practice and rest period. So 10 seconds for practice, 10 seconds for rest. And they had two key metrics that they used to measure the existence of this micro learning that they called. Micro offline learning, which is the difference between the speed at which they finished the block and the speed at which they started the next block. And then micro online learning, which measured the difference between how fast they started a block and how fast they finished that same block. And what we found, which is really interesting, is that during the early blocks, that are the first 11 in this case, we find that overall, most of the improvements in tapping speed actually came from the rest periods rather than from the practice periods. And this is consistent with what they found in that original study. So to test whether our platform could also investigate sort of new scientific questions, we designed our experiment to test transfer learning from the left to the right hand. And you may not have noticed this, but the two sequences that we used are actually the mirror of each other when you look at what fingers they had to use for each one. So for the first one, for example, you have index, small, middle, and so on and so forth. And then for the right hand, you have the same index, small, middle, and so on and so forth. So we defined that there was, we wanted to investigate whether sort of practicing the sequence with the left hand could transfer to doing better with the right hand as well. And the way we measured this was with two metrics, early transfer and late transfer. Early transfer measures the difference in speed between the how fast they started with the left hand and how fast they started with the right hand. And then late transfer measures sort of the ending speeds. And we defined that there was a skill transfer learning if early transfer was big, meaning that they started faster than with the right hand than with the left hand. And also if late transfer was small, meaning that there were no intrinsic differences between the two hands. And this is exactly what we found. We found that early transfer was high of, on average, 1.2 key presses per second. That was significant, with a big effect size as well. And then for late transfer, it was very small, only 0.13 key presses per second difference on average, and with a very small effect size. So overall, we found similar performance first between the supervised and the unsupervised groups, and this suggests that we can actually use our platform to conduct studies outside the lab, which I think is interesting. Second, we validated our platform by replicating this previous study on microlearning, showing that even short periods of rest can contribute to learning. And then finally, we also were able to use our platform to sort of investigate new scientific questions, and we showed that there could be transfer of learning between the left and the right hand when you do mirrored versions of the same sequence. Now, switching gears one last time, I'll talk sort of more in length about my work on brain-machine interfaces for restoring motor control. And more specifically, I'll talk about my work on explainable AI for brain-machine interfaces. And this work was recently accepted for publication at NeurIPS 2024. Before I dive into the project itself, I wanted to give you like an overview of basic neuroscience for those that are not familiar with it in the audience. This is a neuron, or a type of neuron at least, and we have about 100 billion of these in our brain. So these are the basic unit of basically all the computations, everything that we do. And the way they talk to each other is through action potentials or spikes. And if you stick an electrode close enough to a neuron, you'll be able to see this action potential as a change in voltage that happens in a very fast time scale, just within a few milliseconds. To produce movement, what happens is there's a bunch of connections between neurons. They finally connect to what it's called an upper motor neuron that lives in the motor cortex. This upper motor neuron projects down to the spinal cord and connects to another neuron there that is called the lower motor neuron. That could be more than one. And then they connect to that lower motor neurons is the one that actually activates the muscle fibers. So to produce movement, what you have is you need sort of enough of these activating muscle fibers, and you'll be able to see that contraction that actually generates movement. Now, what happens with people with spinal cord injuries and sort of other similar neurological conditions is that this path is broken in some way, meaning that the neurons in the cortex cannot talk to the muscles anymore. And this also happens sort of the other way around. So receptors in our skin that let us sense touch, pain, temperature, they cannot transmit that information upwards towards the brain as well. Each year, there are about 900,000 new spinal cord injuries worldwide. So this is a big problem. And the motivation for our work is to restore the ability for these patients and other people with similar neurological conditions to interact with the world again. And how do we do this? Well, brain-machine interfaces have appeared as a solution in the past 20 years. And the working principle that brain-machine interfaces sort of take advantage of is that you can actually measure the electrical activity of neurons if you stick electrodes close enough. And so to do a brain-machine interface, what you do is you stick an electrode, hopefully as close to the neuron as you can get. A typical electrode that is used for these studies is the Utah array, which is a little bit creepy, I would say. It looks like a bed of nails. And each of these nails is an independent electrode that can measure from sort of individual neurons. And you can record from about 100 at a time with this version of the Utah array. Then from the raw electrical activity, you extract some features. You pass those features to a neural decoder algorithm. And then with that decoder algorithm, you can predict an output control signal. And that output control signal can be used to control a machine. And this machine can be anything from a robotic arm, for example, as has been done before, but also you can control just a simple cursor on the screen and sort of restore the ability for these people to interact with technology again. I'm going to drink some water. Yeah, cool. So now the fact that to produce movement, there needs to be sort of this very complex series of activations and communications between neurons going from up in the cortex to down in the muscle fibers. And what that means is that there's a very complex and possibly nonlinear relationship between brain and movement. And this has motivated the development of deep learning algorithms that are basically models that use big artificial neural networks to model arbitrarily complex relationships. And they can be used by sort of having the brain input or the neural activity as the input, and then producing behavior prediction, such as moving the hand in space. Deep learning, like BMIs have progressed a lot in the past few years, and I think deep learning has played a huge part in that. What I'm showing here is just one example. This is showing a paralyzed participant that has brain implants, and this participant is actually imagining handwriting characters, so like A, B, etc. And then his neural activity is passed into a deep learning algorithm, and then that algorithm is predicting what character that person was trying to write at any given point. And you can see that using that system, he can write pretty fast, and this can sort of restore the ability to communicate at more normal speeds for people that have paralysis. Now, this video is a little bit less scientific, though it's also a paper, so for context, this is a participant that has ALS, and because of that, he has lost the ability to speak intelligibly. It's very hard to understand him when he talks. So what this group at UC Davis did is they implanted electrodes in his brain, connected his brain activity into deep learning algorithms, and they can determine what phoneme he's trying to say at any given point. And with that, they can convert back sort of into words, and he can talk again to people, to his family, caregivers, etc. And this video is the first time that they tried the system with the participant. And I'm just going to play the video because it's really cool.
The first time you tried to use the system, it just worked. Okay, ready? That first time that he tried to speak and that word appeared on the screen, he started crying, I started crying, his family started crying. Basically everyone, we had to pause. We stopped the rest of that block of experiment trials. Everyone composed.
Yeah, so that video is amazing. I cry every time that I see it. And this really shows sort of the potential for really changing someone's lives that this technology has, sort of from being trapped in his body to being able to communicate again with his loved ones at a reasonable speed is such a huge sort of 0 to 1 change. One issue with these applications, the one that I showed just now in speech and the one before in handwriting, is it's not a big deal if the algorithm sort of misspells a word, but when you start interacting with the physical word with the physical world, that brings its own challenges. And for those cases, something interesting happens where linear and explainable decoders are actually the standard for those applications. And what I'm showing here on the right is just one example. In this case, this is a paralyzed participant that is controlling that 7-degree of freedom robotic arm just by using his thoughts. And the group is using a generalized linear model to predict how he wants to move each of the joints, which I think is cool. Now, why is deep learning sort of not more commonplace in these applications? And I think this is because deep learning is seen as a potentially unsafe black box. We don't know what's happening inside. So if we expose it to potentially unexpected inputs, it can have unexpected outputs. And if we have a participant that's actually interacting with the world through this, that can have pretty serious consequences. Of the linear decoders, the common filter is one of the most popular in the field. The common filter allows you to track a state through time by having some information about how that state that you're tracking evolves over time, the state dynamics, and some information about observations of the state, some sensor observations. This video on the right is just one example that is using a common filter for BMIs. And this is a really cool video, I would say. And so you can see there's some gray connectors on his head. That's how the brain activity is getting sort of out of the brain and into the common filter in this case. And he's using the common filter to control that black contraption you see on the right. And then in this case, also directly activating the muscles in his forearm to be able to close his hand and grab the spoon and actually feed himself again, which I think is amazing. Now, common filters are very popular in BMIs, first because they have good performance compared to other linear decoders, and also because they have a very interpretable structure, and I'll get more into that later. However, one issue that the common filter has, that it has pretty strong assumptions. For example, it needs all relationships to be linear, and it needs the noise to be Gaussian with mean 0. And those assumptions are usually not met in brain-machine interfaces. And what that means is that when you compare the common filter to more pure deep learning approaches usually have a lower error, as we showed on a paper last year. So the question is, can we improve the performance of the common filter for BMIs by adding components of deep learning, but still keeping what we like about it, which is its explainability? A few years ago, Guy Rivach, he's a PhD student from ETH Zurich, and they presented this paper on CalmNet, which is a way of combining exactly sort of the common filter with deep learning. And I think they did it in a very smart and interesting way. I contacted Guy about 2 years ago, and we started collaborating to try to apply, sort of adapt and apply CalmNet for our application in particular. And that sort of converged to that NeurIPS paper that I discussed. Now, the common filter and CalmNet both work in the same way. So to make a prediction, you have information about the dynamics and you have observations. And what they do is you use what is called the Kalman gain as a way to interpolate between those two sources of information. And KG is what I'm referring to as the Kalman gain here. Now, the only difference between the two algorithms, common filter and CalmNet, is how that Kalman gain is computed. So for the common filter, you use information about the state covariance, the observation model, and sensor noise to compute this value, the Kalman gain. Whereas for CalmNet, you have sort of fewer assumptions and you use a series of recurrent neural networks to compute this gain. And because the Kalman gain sort of tells you how much to interpolate between what you know from observations versus how much you know from your dynamics. It's also thought of as a metric of trust of the system. If it's high, it means that it's looking more at the observations, it trusts more the information coming from the observations. If it's low, it means it's looking more at the dynamics, it trusts more what's happening in the dynamics. And we call that nonlinear trust. Cool. So to test Kalmanet, we had a monkey do a 2 degree of freedom finger test, which you can see on the video on the right. What we trained him to do is if he sees a red target on the screen, he moves his index towards that red target. If he sees a yellow target on the screen, he moves his 3 other fingers, middle ring and small, towards that target. And every time he goes to those targets and he stays for a given amount of time, he gets some apple juice. And they really go, you would say, bananas for the apple juice. So what we do at the same time is we can record his finger velocities using that manipulandum that you can see on the right there. And at the same time, we can record his brain activity through electrodes that we implanted in his motor cortex, or the part of the brain that is related to motor output. What we can do, which is, I think, really interesting, is that we can actually have the monkey do the task in what it's called brain control mode, in which we record his brain activity in real time. We pass this through a decoding algorithm, and we can use that to control, for the monkey to control this virtual hand in the screen sort of in real time as well. And in this work, we compared 4 different algorithms, the common filter and Kalmanet, which I already discussed, and then 2 pure deep learning approaches, the TCFNN and LSTM. The TCFNN is a feed-forward approach, and the LSTM a recurrent approach. We first compared how the algorithms did in predicting offline data or pre-recorded. So we had a ground truth, so we wanted to see how well we could match it. And I'm showing just a snippet of that on the left. The blue shows the index velocity, and just a snippet of time of that. And then the different colors represent the predictions by the different algorithms. And you can see that overall, in offline predictions, Kalmanet and the LSTM do the best for predicting those very high valleys and very high peaks and very low troughs. When we aggregate across states, we find a similar effect in which the Kalmanet achieved very similar correlation coefficient between prediction and ground truth to the LSTM. And a similar thing happened with the mean squared error, where they achieved similar mean squared errors. So now moving on to brain control mode. And what I'm showing here is our trajectories of the fingers over time. So in yellow, I'm showing, hopefully you can see that. In yellow, I'm showing the position of the index over time. And then in blue, I'm showing the position of these 3 fingers. And each of those rectangles that you can see in the screen is where the target was. And for the monkey to get that apple juice that they go bananas for, they had to stay within that rectangle for 500 milliseconds. And you can see sort of a very notable difference between the 2 algorithms, in which the LSTM had a lot more oscillatory activity, sort of, as you can see there, and maybe there as well, compared to the Kalmanet, and Kalmanet was a lot smoother in its path. Now here's a video of how the monkey controlling the virtual hand actually looked like for the LSTM on the left and Kalmanet on the right. And what we see sort of overall is that Kalmanet was a little slower than the LSTM when doing these trials, but it was smoother in its path and also more successful at acquiring these targets within a desired time frame. Desired time frame. Now, how did Kalmanet achieve this good performance, right? We care about explainability, like I've said that word many times during this presentation. So if we can't explain how Kalmanet worked, we would just be monkeying around. So to do this, we looked at how Kalmanet modulated the common gain over time and compared that to the finger velocity. So in black, you can see on the plot that I'm showing to the left, in black, I'm showing the finger velocity over time, just a snippet. And then in orange, I think orange is the proper color. Orange, you can see the common gain coming from the common filter. And then in green, the common gain coming from Kalmanet. And you can see that for the common filter, the common gain stabilizes very fast within just a few seconds, which is a sort of a known thing that the common filter does when it has a constant noise model. whereas Kalman had a lot more interesting behavior in which its Kalman gain covaried almost perfectly with velocity. Now, this makes sense when we look back to how the Kalman filter actually works. So the Kalman filter, to produce a prediction of the finger velocities, it interpolates between the values that you get from the dynamics and from the observations, right? And in this case, our observations are the brain activity. Now, again, so when the Kalman gain is high, it means that it's looking more at the brain activity, meaning that Kalman looked at the brain for producing high velocities. And then when it's low, it's looking at the dynamics, meaning that it looked at the dynamics for stopping. So overall, we see this nonlinear thrust modulation of the Kalman through its Kalman gain. And something interesting is that this may tell us something cool about the brain, that it may be more information rich during high velocity movements, which may have been the reason why Kalman sort of chose to look at that for those predictions, than during a steady state in which it may be less information rich. We wanted to test sort of that this is for sure the mechanism through which Kalman achieved its good performance, this modulation of the Kalman gain with the velocity. So we built another model called the HKF, or heteroscedastic Kalman filter, which is basically a linear Kalman filter with the only change that we switch how we compute the noise model for the dynamics. So instead of having a constant noise, we have now a variable noise, and we designed that noise model so that it has a similar behavior. So the Kalman gain has a similar behavior to what we see with the Kalman. Interestingly, we found that when doing that only change, we can match the performance when predicting finger velocity with Kalman. We can match that with the HKF, which is just a linear Kalman filter with a variable noise model. And you can see that both in terms of correlation as well as mean squared error compared to the Kalman filter. Now, it's not all good. What we see is that Kalman suffers a lot under noisy outliers. So what I'm showing on the plot to the left is as we increase the noise magnitude, what happens to the mean squared error of the prediction for each of these models. And you can see that as you increase the noise magnitude, Kalman does really, really bad very fast. And then something similar happens, but to a lower extent, when you increase the noise duration, which is what I'm showing on the plot to the right. Something very interesting that we found here, which we didn't want to find, was that the LSTM did actually a lot better at rejecting noise than Kalman and all the other models did. Now, this is sort of, it makes sense from the Kalman filter perspective, because a known weakness of the Kalman filter is that it's really bad at rejecting noise outside of the model distribution. And so this makes sense, but it was interesting to see that the LSTM did so well when injecting noise that was many times what he had seen during the training data. So that was pretty cool. And this may mean that the LSTM may have sort of an underlying or an implicit model of the noise distribution on the training data, and potentially also like an outlier rejection mechanism, an implicit outlier rejection mechanism. So overall, we found that we may not need to sacrifice explainability to achieve good performance. And this work highlights, I think, 3 benefits of explainability. First, when you have an interpretable structure, you can get insights on the input. And we see that with our work, where we found that the brain may be more information rich during high velocity movements than during sort of more steady state. Second, when you have an interpretable structure and you also have good performance with the model, you can get insights on model development. And we did that by developing this new sort of linear model, the HKF, that did the same mechanism that Kalman did, and we can match the performance with just a linear model. And then finally, when you have an interpretable structure, but you have bad performance, that potentially means easier debugging, because if you know what each part of the system should do and you know what's failing, then you can potentially do you have an easier job at sort of correcting those errors. With all that monkey business done, I wanted to finish by showcasing the publications that I've had a chance to be a part of. First, as a first author, I've had 3 papers plus one that it's in its very final stages, hoping to submit by a few weeks. And then also as a co-author, the chance to be in 2 labs has allowed me to work on many interesting and very cool projects, and I'm very grateful for that as well. Cool. Okay, So now to acknowledgements. First, I wanted to thank my committee for being here. I really appreciate you all, Cindy, Elliot, Brent, and also Muli on Zoom. And I really appreciate all the guidance that you have given me, especially during the last few months. And thank you for taking the time to do all this as well. Cindy and Muli, I wanted to sort of more especially thank you for the guidance that you have given me as advisors more specifically. And the work that I've had a chance to do during my PhD has really sort of driven me closer to what I want to do and the goals that I have. So I really have to thank you for that. So thank you. And then also, Cindy, I wanted to more specifically thank you for that lab culture that you have in your lab is really, really cool. It's nice and very cool. It's nice to go to work every morning and actually like your lab mate. So I really appreciate that. I also wanted to thank the robotic staff. I just highlighted a few names here for all their help during the last few years. And I wanted to more specifically thank Denise because she has been an amazing part of this. She has supported me so much through many different processes. So you're the best, Denise. And then I also wanted to thank all my friends for both the ones in the lab, my lab mates, Eric, and all the ones that I've met in Ann Arbor, and also all the friends back from Chile. You have made this past 4 years an amazing time. And this is just a collection of photos that I've had with some people. I'm missing so many more, but I really wanted to thank you. Thank you for making this such a cool time. And then I wanted to thank my family. I have a very big family, both the ones that I was born into and the one that I chose through Sophia. I love my family, both of those. And I wanted to more specifically thank my parents that are here today, and they braved the Ann Arbor cold, and they flew a long distance to be here. And this would not be possible without you. So just thank you. And just so you get a sense of how big my family is, this is the extended, like the nuclear family of Sophia's and I's combined for our wedding. So it's a lot of people. And then finally, but definitely not least, I wanted to thank Sophia, my wife. You're the best. You have been there for every step of the way. You've been my biggest fan when things were not going, when they were going well, and then my rock when they were not going well. And first supporting me from the distance from Chile, and then you came here sacrificing a lot to be here with me. You are amazing, the best person I've ever had the chance to meet, and I'm looking forward to the next 65 years or so of our lives together. If I get the PhD today, you deserve it definitely more or as much as I do. So thank you, Sophia, for everything. And with that, I wanted to thank you all, and I'll take any questions.